Hi, everybody. Today's Monday, October 12th, and I wanted to extend our conversation on LTP and LTD into some practical examples of how these things occur in our everyday lives. I wanted to give you also some examples of experiments that were performed in the 50s through the 70s. And I keep showing you these videos because these are the only videos that exist. A lot of the experiments that were performed at that time are actually not considered to be ethical right now. But what we'll do today is we'll talk about fear conditioning and fear extinction, which exhibit forms of LTP and LTD, though I'll try to touch on a little bit today the controversy surrounding why these things may or may not be fully attributable to LTP and LTD. And then we'll talk about the neurocircuitry of addiction. I just want to give you the basics today so that you understand sort of basic applications of LTP and LTD. And then when we move forward on Wednesday, we'll talk about a little bit more sophisticated systems like depression and anxiety and how those things affect learning, which is a form of LTP. So this is our schedule. We are now in the third week of module 3, which is changing neuronal connections. We talked about experience-dependent plasticity, which is plasticity that occurs either prenatally or early in childhood development. And we talked about the molecular mechanisms of synaptic plasticity in the adult. And like I said, this week we're going to talk about some applications of these things. There is no quiz next week on practical examples because this week's content will be looped into Friday's exam. The exam will be open at 11:30 a.m. like we did last time on Friday, and it'll close at 11:30 a.m. on Saturday. You'll have 60 minutes to complete that exam, and although I haven't finished writing it, I anticipate it also having 6 questions like it did last time. Okay, so let's take a quick look at the limbic system. So we've talked about a number of different brain regions already, but we're going to move a little bit deeper in the brain. First, I just wanted to give you sort of an orientation to the regions that we're talking about. So there is a way of categorizing portions of the brain into the neocortex, the limbic brain or the limbic system, and the reptilian brain. And each of these components of the brain are responsible for different things. For example, the neocortex, which is the superficial layer of the brain, is thought to be responsible for reasoning through problems and thinking, the way that we think about our brain functioning every day. The limbic system is also called the feeling brain because it's in the limbic system that a lot of the regions are located that are responsible for emotion. And we'll talk about this later today, but the idea of emotion is critically tied to things like motivation, learning, addiction, and habits. So the limbic system itself, the limbic brain, is fairly complicated. It's comprised of a number of very tiny regions, and we'll talk about a few of them. And the reptilian brain is also called the dinosaur brain. This is mainly in the brainstem, and it's responsible for instinctual behavior and survival. So things like the drive to eat, to have sex, to reproduce, and basic things like breathing and heart rate are regulated by the brainstem. In this course, we're not going to talk much about the brainstem at all until we get to the spinal cord. And even then, we're not going to really talk about its specific integration into the rest of the body. But I just want you to know that the brain stem is where a lot of your basic function resides. So for example, if there's no brainstem activity and you're on life support, well, you would be on life support because you wouldn't have heart rate or breathing regulated by the central nervous system. But that level, if there's activity there, the person is not technically considered brain dead, although brain dead can also be defined as cortical inactivity. Well, let's look a little bit more closely at the limbic system. The limbic system is very closely tied to the hippocampus. So you've already seen the hippocampus. That's located here. And the hippocampus is the thing that sort of looks like a seahorse. And this is where we've been focusing our conversation on LTP, because this is where it's the best studied in those CA1 and CA3 neuronal connections. And we talked about the role of glutamate, NMDA, and AMPA receptors in that region of the brain. We're going to expand that a little bit to consider the thalamus. Now, you have already talked about the thalamus. The lateral geniculate nucleus is located in the thalamus, and the thalamus itself serves as a relay station for a number of different systems like vision and hearing and smell. It's thought to sort of organize information before information is sent to the cortex for higher processing. Now, the amygdala is one structure that we're going to be talking about quite a lot. The amygdala is located sort of at the tip of the hippocampus. It's just forward of the hippocampus. And remember that we're using these terms as singular, but the amygdala exists on both hemispheres of the cerebrum. So you actually have two amygdalas, amygdalae. But in any case, they're located just to the front of the hippocampus. So it's in these regions of the brain that conditioned learning takes place, mainly through the interactions that are occurring at the amygdala, which also exhibits LTP. So I'm going to show you first a video on a child. The child's name is Albert. And this was an experiment that was done, I think, in the 50s. And it's called the Little Albert experiment. I want to emphasize that you would not be able to do this experiment today. But let's just take a look at what fear conditioning is. It's basically making the association between something that shouldn't elicit any real response and connecting that to something that elicits a behavioral response. So the classic thing is, you become scared of something irrationally because you had a bad experience, and your brain associates those two things together. This is conditioned learning that's very similar to Pavlov's experiments. So the experiments performed by Pavlov in dogs, where the dogs started salivating because they hear the bell, those experiments are tied into the amygdala and are using the mechanisms that we've been talking about. So let's look at this very poor quality video of little Albert in the experiment that was performed. So I don't think there's any sound associated with this. But basically little Albert, I think he was 9 months old. And what they're doing is they're showing him cute things. Now, I don't know how this was okay, but anyway, they're showing the baby a monkey, and the baby's like, oh, wow, there's a monkey. That's kind of neat. And the monkey is furry. That's the thing that you should be paying attention to. And they bring over a dog, and the baby's like, oh, look, another furry thing. It's kind of a strange looking dog. But anyway, the baby shows interest in it. It's only mild interest, but it's like, oh, wow, look at that. Then they bring over a rat. Now, the choice of animals to me is bizarre, but the baby is interested, right? You can see the baby reaching out. The baby's not afraid of the animals. So what they're going to do is they're going to present the baby with a very loud sound at the same time that the baby sees the animal. And these two stimuli together will create a conditioned response that will make the baby afraid of the rat. So they don't show the pairing. I have seen a video of it, and it's a little bit horrifying. But not only is the baby now afraid of the rat, which was the training period, but now the baby can't tolerate anything with fur. That conditioned response in this baby's brain, in Albert's brain, is transferred to anything furry. I think because this child hasn't seen anything that's furry before. This is a child who wasn't exposed to any animals. And the thing that was most, okay, that's just scary, right? That's scary. That's ridiculous. Anyway, But it was a way to explain how somebody might be afraid of something when there isn't any reasonable reason to be afraid of that object or that animal. Now, more recent experiments are a little bit better controlled, and they're performed in rats. So what you're looking at here is a rat that's pressing a bar. You can see that. And the bar pressing is associated with a tone. So the animal is hungry. They've probably food deprived this animal for at least 24 hours. And what they're doing now is they're having the animal press the bar to get the food, but now the tone is going to be paired with a shock, and the animal is going to become afraid of the tone. And you'll see that in just a second. The way that rats exhibit fear is through freezing. So if you're interested in doing behavioral studies in rats, it's a little bit boring because you just watch to see how long the animal freezes for. So see, they show the tone, or the animal hears the tone and he goes, "Ah, something's happening." And he waits, freezes, and then he'll go back to what he was doing. But he becomes afraid of the tone, which is not reasonable. Now they're saying that this isn't, it's a shock on the feet that shouldn't be producing any real pain. They used to do it in such a way, and I'll show you a video where the animal is obviously in pain, but this is supposed to be just unpleasant, which I can imagine, it's easy to imagine. So the pathway that has been proposed for being responsible for associating, say, sound with fear, and then transferring that fear of the sound to something else, it involves various areas of the brain, including the amygdala, and they all kind of converge onto the amygdala. So you hear the sound, it gets transferred to the medial geniculate complex, that's in the thalamus. It gets processed in the auditory cortex and is sent to the amygdala, or it can go directly to the amygdala. Now, what's been a little bit controversial is that these ultimately converge onto motor activity. Somatic motor activity is the motor activity that we have control over. Visceral is your gut, which is, you know, the literal interpretation of that is you were so scared you shit your pants. That's because the brain is controlling also your visceral nervous system, the enteric nervous system. We can talk about that sometime if you're interested. But we're going to sort of keep this focus on this pathway. So one of the debates about this type of learning is not that it involves the amygdala very much at all, that maybe it's something that's happening at the level of the neurons that are controlling the motor areas, especially in the rats that are freezing. So this at this point in these early experiments, the involvement of the amygdala in these pathways were somewhat controversial because it was thought that the freezing that was measured really was a motor response and didn't really have much to do with anything happening in the amygdala. Now, in more recent studies, and this one is probably 20 years old now, people started implanting electrodes to see if there was actually learning taking place in the amygdala, that the amygdala was starting to hardwire associations between things that before didn't elicit any response at all, like the furry animal or the tone. So you can do this experimentally using electrophysiology. What you do is, we've seen these experiments before where you drill a hole into the skull and you drive electrodes into the brain. And you don't do this willy-nilly. You look at a map and you measure relative to the sutures on the skull and drill the hole in the appropriate places. And here what they're showing is that they are able to stimulate in the hippocampus, and they can elicit responses in the amygdala. So the amygdala is the dark gray here. And then when they do that at high frequency, in the same way as you would when you're eliciting LTP in the hippocampus, you can measure increases in responses over time because of that training and because of LTP occurring in the amygdala. So the point of all of this was to demonstrate that LTP isn't really localized to just the hippocampus. It occurs in a lot of different areas of the brain. And one of the areas of the brain that's really important is the amygdala. People knew that this area of the brain was responsible for emotions, but they didn't realize that emotions are so tied into our daily functioning that you can actually have effectively conditioned responses or learned emotions associated with things that normally wouldn't elicit a response. And that is literally LTP. In the same way that LTP can be interrupted in the hippocampus with protein inhibitors, you can do the same thing in the amygdala. So this requires not only short-term changes, but also longer-term changes that involve gene expression. So just to remind you, the way that that occurs is through the release of glutamate. So just like in the hippocampus, the learning or LTP that takes place in the amygdala also involves glutamate binding to NMDA receptors and AMPA receptors. And the primary effect of binding to those receptors is an increase of postsynaptic intracellular calcium and sodium. And the calcium does a number of different things. One of the things that it does is it activates CAM kinase 2, and which phosphorylates AMPA receptors that relocalize to the synaptic terminal, so that they are poised and ready to receive information, and the concentration of receptors is increased where things matter. So that's really just a transient effect, because remember, LTD removes the AMPA receptors. So that can it can be reversed pretty quickly. And likewise, PKC can also affect different channels or receptors that will influence the depolarization of the postsynaptic membrane. So there is also an effect called extinction. And extinction is basically the removal of that fear. You know, I at one point was pretty irrationally afraid of bugs. I don't know when it happened in college. I don't know what started it. And then it just kind of went away. So in some way, I probably had some conditioned response to bugs that involved a huge amount of fear, completely irrational, not bugs that could kill me, but just like ants. And then extinction occurs where it goes away. Now, the extinction of fear on a molecular level or a cellular level is, again, somewhat controversial. So there are neurons that are receiving the conditioned stimulus, and that afferent projects to what's called a primary neuron. And that primary neuron is the thing in the amygdala that causes fear. This neuron, the sensory afferent that receives the conditioned stimulus, whether it's sound or light or pain, these afferents also connect to interneurons, which is indicated as an N. And that can influence this connection here that's circled in blue. There are three, that's conditioning. The extinction of fear requires probably one, if not more, of these three mechanisms, where you have a depotentiation, where it just goes away because this isn't as active anymore, and then you get a lower amount of fear. Now, extinction can be reversed very quickly, but that's called depotentiation. And if you're looking at this thinking, I don't understand how that happens, it is just as sort of fishy as it looks, where there's potentiation at a neuron here, LTP, and depotentiation just means that kind of goes away. It's kind of a crappy explanation, to be honest. And then there could be LTP at the interneuron. And this is a different way of thinking about LTP, where this synapse between the sensory afferent and the interneuron is strengthened. And because it's strengthened, it means that there's an increased probability of that interneuron in firing. But instead of firing in a positive way, this is an inhibitory neuron. So this synapse that causes the interneuron to fire is more efficient, and therefore the inhibition that converges onto this primary neuron is increased. And the way that the inhibition is achieved is through the release of GABA. Likewise, this connection at a high frequency stimulating this neuron can cause potentiation at this synapse. So this is inhibitory LTP, where the LTP occurs between an inhibitory interneuron and a primary neuron. So just to review, because I know this is just a lot of terms being thrown out, but if you think about it, there really aren't a lot of places here that we can change. This is a simple circuit, and there are only 3 areas. One is where the synapse was potentiated to begin with. The second is causing this neuron to fire more. And the third is to cause this synapse from the interneuron to be more efficient, to cause potentiation. So LTP can exist at excitatory synapses and also at inhibitory synapses. The excitatory synapse would be responsible for the development of conditioned fear responses, and then the potentiation at an inhibitory synapse would be responsible for the extinction of that conditioned fear response. Now, another mechanism that I think has also been demonstrated much more recently than that schematic was created is the idea of LTD happening. You saw this at the hippocampus, and these mechanisms have also been shown to be in place in the amygdala now. So activity at a glutamate receptor, but rather than high frequency activity, low frequency activity can activate protein phosphatases through the influx of calcium that causes the relocalization of AMPA from the synapse back into the endosomes. So it's reducing the efficiency of that synapse. And this has been shown both in animals and also to some extent in people. So fear conditioning and fear extinction are mechanisms that, or the mechanisms causing them are ones that are similar to LTP and LTD. But what I want you to remember is that you could interfere with any of those synapses pharmacologically to decrease the efficiency of those connections and decrease conditioned fear. There's a very fine line between conditioned fear and anxiety, and I want to talk about that on Wednesday, and we'll also talk about some medications that are used to control these things. So if you're thinking now, I don't get it, we'll cover it again on Wednesday, and then on Wednesday we're going to talk about whether you'd like to have a review on Thursday for the exam on Friday. So let's introduce a little bit addiction. I have stress and plasticity on here, but we're going to move that to Wednesday. So addiction also involves the limbic system. The amygdala is involved, the thalamus is involved, the hippocampus is involved. The amygdala serves a really important role in addiction because there's a huge emotional component to addiction, both to chemical substances like drugs and also to food and sex. These things can't be separated from emotion. People enjoy food, people enjoy sex, and it's not surprising that this system is also involved in addiction. So I'm going to show you some early experiments in humans that involved localizing the area of the brain that might be responsible for pleasure and how that ties into people's need for pleasure. Because addiction really is about emotion, but unfortunately it becomes hardwired in the addicted brain, which we'll talk about the mechanisms in just a minute. I'm going to fast forward through this because there's like a stupidly long introduction. Here we go. I don't know when this is from, but I'm going to guess the 1970s.
You almost look like you're smiling a bit. Are you?
Oh, no, I'm not.
Heath implanted electrodes in the brains of chronically depressed patients, hoping to stimulate pleasurable sensations.
I guess I've cracked up all the way. I don't know.
You mean that's the reason you're smiling?
Probably.
Well, you look like you're real. What are you laughing
I
about?
don't know.
Huh?
Are you doing something to me?
Not surprisingly, the experiments were controversial, but the electrodes did have an effect, especially near an area of the brain called the nucleus accumbens. Heath even let his patients control their own stimulation.
I find this button the best.
That's number 2 button.
Most pleasurable.
How do you feel?
So here this woman Just a little bit of background on her. Yes, she was a chronically depressed patient, but she was also diagnosed with schizophrenia, and she was institutionalized. So she was in a psychiatric hospital in a pretty permanent sense. She also didn't have any family, because you can imagine that if I said to you, hey, I hear you've been depressed for a long time. I was thinking I might just throw some electrodes in your brain and just stimulate different areas. Maybe I'll put them here, put them there, put them in another place and see what feels good. You probably wouldn't consent to that. So these experiments are no longer done like this, where they do like just a look-see. But in any case, they were able to identify the nucleus accumbens as an area of the brain that not only made people feel happy and experience pleasure, but also an area of the brain that made them want more. Because it's one thing to, be out in a field of flowers and feel really peaceful and happy. And it's another thing to compulsively just want to be around those flowers all the time.
I made a small joke yesterday. I don't know if I should repeat it or not.
I'd like to hear it.
Do you think it's worth repeating? What about the very deep, deep electrode all the way down?
You're trying to tell me how it excites you, is that it?
No, I think it's kind of I think it's somewhat of a
She has a
button.
choice at this point of multiple electrodes that are
Well, you said it went all the way down.
implanted in her brain, and she's choosing the
Perhaps
one
we get pleasure
now
from
that's
drugs
in
because they
the
stimulate the same
nucleus
brain
accumbens.
areas as sex and food. Working in Montreal in the 1950s, James Olds devised a crucial test. He worked out a new way to, in effect, ask animals how they felt.
This is a device which trains the animal to turn on the stimulus for himself. Here you notice the animal wandering toward the pedal. When he first touches it, he gets no stimulation.
So these experiments are done today in the exact same way. So I told you about using dental cement and implanting chronic electrodes onto the surface of a brain. So what the experimenter has done here is taken a tiny Dremel and drilled a hole just to the surface. And we saw this in other animal experiments already. He cut a hole in the dura and then drove electrodes into the brain. And what they're doing now is they're going to train this animal very, very quickly to press this button to get stimulation of the nucleus accumbens. And you'll be able to see that with a light that simultaneously goes on when the animal's brain is stimulated. Now, this is the first time the animal's seen this apparatus, so you'll see the effect. It's dramatic how quickly it happens.
It's not connected yet. Then one time he touches the pedal, it does turn on the stimulation. You can see the lights go on.
Now, the interesting thing about the old stimulation, the rewarding stimulation, was the strength of it. It was somewhat different than rewarding stimulation for food and water in that there was no satiation. Animals would continue for hours and hours and didn't seem to satiate. They didn't feel full. They would just keep doing it for long, long periods of time. They would do it to exhaustion.
Now, I will electrify the grid. The animal must cross an area that gives a very painful shock to the feet in order to get to the pedal and stimulate the brain.
And a rat wanting brain stimulation would brave a shock stronger than even a starving rat would brave to get food.
This shows a rat is willing to pay a very high price in order to get to the pedal.
Human addicts will pay just as high a price for their rewards.
Yeah, so they haven't done this kind of experiment with humans, but I think that this kind of experiment occurs every day in people who are addicted to various chemicals, whether it's alcohol, cocaine, heroin. Loads of drugs are addictive, as you know, and people will nearly kill themselves to have the drug, regardless of what the effect is on family or on their livelihood. It's interesting to me that this is such a powerful system that gets hardwired into the brain, and it's really, really hard to get extinction of that. But if you're interested in this kind of research, in the 1970s, there was an experiment, and I'll try to find a video of it. I haven't been able to find, but I bet there's a documentary I can pull a clip out of. If you've heard of Rat Park, there's actually a really great documentary called Rat Park that was done maybe 10 years ago now. And this was a psychology experiment where instead of giving a rat the choice of two bottles, one of which just had water and the other one had heroin, they put the rat into a different environment. So if you just put a rat into a cage and you give them water or water with heroin, they will keep drinking that heroin until they die. And that also occurs in people, right? If you give people enough heroin, they'll keep doing it until they're dead. But if you put the rat into what the psychologist called Rat Park, which was basically the most awesome rat cage, it had toys, it had friends. A lot of rats at this time were housed individually, and rats are very social animals, so it was really a deprived environment for them. But if you then give them friends, literally same-sex animals that they could just hang out with, and opposite-sex animals where they can have sex, they're happy and they choose not to drink the heroin. So if you put the same bottles into Rat Park, it's something like only 10% of the animals will drink the heroin compared to 90% of animals. And they haven't really figured out, to my knowledge, the difference between the animals that choose to drink the heroin and can't stop and the animal that can resist. I know that there's been some work on it, but I don't think that there's a definitive answer on that yet. Now, the addiction circuitry is very similar to the circuitry that we saw in fear conditioning. And on a molecular level, addiction is very similar to LTP in the sense that the synapses are changed and there's coordination between not just two neurons, but also others that come in and start to modulate either the synapses between the two, or they make synapses on the same neuron to strengthen the response of the postsynaptic cell. So there have been, you'll see in your reading that there are 3 different sort of phases of addiction, but each of these involves the ventral tegmental area, the VTA, the prefrontal cortex, which is located here in green, the nucleus accumbens, which is very close to the amygdala. This is what they're calling the extended amygdala in red. And these areas are all in the middle of the brain, and they're all super interconnected. So there are loads of different responses that can be elicited, and because each one of these neurons receives tens of thousands of connections from other cells, these connections can be very powerfully reinforced. In this area also, there are cells that are responsible for food drive, sex drive, pleasure. So this is almost a dangerous area for the kind of lifestyle that we all live, where we have access to loads of things that give us so much pleasure, that stimulate the amygdala and the nucleus accumbens. Video games are another one that stimulate the nucleus accumbens. So, you know, when people say that they're addicted to social media, to video games, to cocaine, it's all really the same circuitry. So if we just look at the effect that different drugs have on these connections between the ventral tegmental area, this is the projection neuron that connects to the nucleus accumbens. And you can see that these are depicted as they're really spiny. They have tons and tons of processes because they're just taking in a ton of information from different areas of the brain. That you can have an interneuron that connects to the ventral tegmental area projection neuron that inhibits. And if you think about things that are considered depressants, opiates and alcohol affect these cells and the synapses they make onto the ventral tegmental area neurons. There are also cells that release glutamate to excite these cells, and those cells, the synapses of those cells can be stimulated by nicotine. So nicotine has the same effect as stimulating this circuit that produces pleasure. So although nicotine doesn't have very many physical very many physical effects. What we would call psychological effects are really powerful, because the nicotinic synapse here, although there are nicotinic synapses everywhere, you feel it most in the brain and in the nucleus accumbens. There's also cocaine and amphetamines. These can affect, and we'll talk about this later when we talk about dopaminergic synapses, but they affect the way that dopamine acts at the synapse, how much is released and how much is taken back up by the presynaptic cell. So the effect of the dopamine, which directly stimulates the nucleus accumbens and is thought to be one of the most important neurotransmitters in feeling good, is left in the synapse for longer. And again, opiates and cannabinoids affect this system also. So cannabinoids are different from alcohol in the sense that they will directly affect the nucleus accumbens. So when people are using cannabinoids, they often feel really good. People who are taking alcohol will often feel good or feel really bad, but it depends on the rest of the circuitry in that case. Now, when the circuit is addicted, what happens is there are changes in gene expression. So if we look at the glutamatergic synapses here, these are the neurons that are releasing glutamate. You have changes in the postsynaptic cells that will decrease dopamine, will decrease glutamate, will increase glutamate receptors like the mGluR1 and the downstream signaling molecules like tyrosine hydroxylase, which you know is involved in dopamine production, and CREB, which changes gene expression in response to glutamate. And there's increased cyclic AMP and CREB in the nucleus accumbens. So what happens here is, because of the chronic stimulation of these synapses, the system says, I don't need to do this anymore. This is nuts. And it decreases the amount of glutamine produced. It decreases the amount of dopamine, and people feel really bad. They feel physically horrendous when they stop taking these drugs, because instead of going back to baseline, they've gone lower than baseline. So their nucleus accumbens, if you think about that being sort of the happy area of the brain, is really not capable of producing pleasure without an enormous amount of stimulation. And without that enormous amount of stimulation, everything that's responsible for it has been downregulated. So it's really, really hard for people who are physically addicted to drugs to get off for these reasons, because of glutamate signaling, dopamine signaling, CREB and cyclic AMP, and also the expression of glutamate receptors. So I want you to understand from all of this, from the discussion of addiction, which will continue also on Wednesday, is that the drugs themselves can physically change not the way that the cells connect to each other, but the efficiency at which the connection takes place. So this, you could think about this as a form of LTP. And we'll talk about this a little bit more on a molecular level and how decreased glutamate affects the structure of the postsynaptic membrane in the addicted brain. Okay, so we have gone through fear conditioning and fear extinction, and we talked a bit about addiction. So as I said, the goal today is to just give you the basis for discussing fear conditioning and extinction in the context of LTP and synaptic plasticity, and also the types of molecules that are responsible for producing pleasure and then for downregulating that when there's too much stimulation because of the presence of a chemical. So I'll see you on Wednesday. I have office hours tomorrow from 1:30 to 2:30. I also have office hours at 12:30 on Friday. So swing by if you have any questions, and we'll decide tomorrow if you guys want to get together or if you just want short tutorials or a mini review session that I can just record for you. Okay, I'll see you then.